So it's my pleasure to introduce two Morgridge scientists. Tony Gitter is an investigator in virology, and he is an assistant professor in the Department of Biostatistics and Medical Informatics, and is a member of the Cancer Center, the Carbone Comprehensive Cancer Center. Paul Ahlquist is a investigator at the Morgridge Institute, and he directs the John and Jean Rowe Center for Virology Research at the Morgridge Institute. He is a professor of molecular virology and member of the Cancer Center, and he is an investigator at the Howard Hughes Medical Institutes as well. So our approach to our program today is we're going to cover some important aspects of the COVID-19 pandemic and the underlying virus that causes it. And I'm going to encourage everybody to enter questions in the chat function on the screen. Our plan is to go ahead and address those questions later on in the session. So the Morgridge Institute was started in 2006. We came to our current physical location on the campus in 2010. Paul Ahlquist was one of the initial scientists at the Morgridge Institute, and so our scientific programs have been addressing questions of virology ever since the Institute started. I think it's important that we think about this pandemic that we're facing right now in the broader context of viruses and how they enter into our society and then how we respond to them. And that's the purpose of this webcast. So let me start off by introducing Paul Ahlquist to you. And Paul, let me ask you to summarize your research and your background.
Well, thank you, Brad. I'm fortunate to be working with a truly outstanding group of researchers in our lab. And they're studying the molecular mechanisms that viruses use to replicate and to interact with cells and to cause disease. Our studies span a range of different viruses, and this includes, for example, HIV and two major cancer-causing viruses, human papillomavirus and hepatitis B virus. We also have a major effort that's been long-standing that's uncovering the central mechanisms in genome replication for a very large class of RNA viruses that includes the coronaviruses, Zika virus, paralytic poliovirus, and many other important pathogens. And I hope to say a few words about that work later.
Thank you. Tony, if I could ask you to introduce yourself and tell us about your area of research and your background.
So I lead a computational research group at the Morgridge Institute, mostly focused on virology, and I lead a group of computer scientists and statisticians and engineers who are developing new software tools, machine learning tools to analyze the data that comes from biological experiments. So our goals are to think about new ways to analyze that data so that we can help our collaborators like Paul and many others look at it in new ways, maybe think of different follow-up experiments that we might pursue, especially within different virology applications and cancer-related viruses.
Great. Thank you very much. I'd like to start off with a couple of definitions. And so first, coronaviruses. So coronaviruses are an entire class of viruses. And you've probably seen pictures of the virus or artist's rendition of the virus that is responsible for COVID-19. And it's got all these spikes sticking out of it. And because of the appearance of the virus, it sort of looks like a crown. And that's where the term coronavirus comes from. So it's an entire class of viruses. The original virus that caused diseases similar to COVID-19 was called severe acute respiratory syndrome, or SARS. So the SARS virus is a coronavirus. And then there was a second virus that emerged, and it came from the Middle East. It was called the Middle Eastern Respiratory Syndrome virus, and that was also a coronavirus. The new virus, the virus itself is called SARS, which again is severe acute respiratory syndrome, dash CoV for coronavirus dash 2, because it's the second coronavirus that causes severe acute respiratory syndrome. And so that's the virus. The name of the disease is COVID-19, for coronavirus disease, in 2019 is when it started. So COVID-19 is the disease, SARS-CoV-2 is the name of the virus. So rather than recap what we know about SARS-CoV-2 or COVID-19, we want to put this whole set of events into the larger context of newer viruses seemingly coming out of nowhere and then catching us by surprise, and how do we react? So Paul, let me ask, what was the reaction in the virology community when SARS-CoV-2 began to emerge and we saw it evolving into a pandemic?
Well, I think first and foremost, the virology community, just like the general public, quickly began to realize that this was the public health event of the century, and that it demanded a massive response in all spheres, across public health and medicine and the economy and other areas. The second parallel reaction was that this pandemic was exactly what many researchers and public health officials have predicted for years. For example, in our campus virology course, I've been teaching for over 10 years that pandemic events like this would happen and discussing their likely course. Now, we knew that there would be such pandemics and that yet others will occur in future, because the COVID-19 pandemic is not just a unique incident. It's just one step in a series of dangerous virus emergences from before the 1918 flu, through HIV, through recent outbreaks of Ebola virus, the earlier SARS and MERS coronaviruses that you mentioned, Brad, Zika virus, and many others. Now, all of those that I mentioned spread internationally to varying degrees. And in each outbreak, the virus is trying to use its enormous mutational capability to adapt to spread even faster and further in humans, as SARS coronavirus 2 has done particularly well in this pandemic. We also have to expect future pandemics similar to COVID-19, since the emergence of dangerous new viruses like this is actually accelerating for multiple well-understood reasons, including the fact that the human population density is increasing significantly. We have increasing encroachment on wild habitats that are the sources of these new viruses, increasing international travel and trade, and so forth. So again, this is a continuum that has preceded COVID-19 and will assuredly extend beyond it.
That's a very important point. And I think that certainly SARS in 2003 and MERS in 2012 got our attention, but it did not turn into the global pandemic that we're seeing now. And COVID-19 certainly has our attention. So there's a temptation then to focus just on what we have to deal with now. And in the big picture, should virologists just focus on the coronavirus that we're dealing with now, or should our focus be broader?
So there's two ways that we should think about this. On one hand, in the very short term, we are living in the middle of an ongoing pandemic. So certainly virologists and many types of scientists should be thinking and are currently thinking about how can we use the best scientific tools that we have available to end this pandemic as quickly as possible. How can we develop vaccines? How can we develop treatments against this specific coronavirus that's emerged? But then thinking back to what Paul said, we could not predict that this would be a coronavirus that had such a global effect. Yet virologists do expect that under current conditions, we will see future viral pandemics emerge. So without the ability to know whether it will be influenza, coronavirus, or something else, it really does demand that as virologists think about what the next steps are and continue their ongoing work even in this pandemic toward those next steps, we need to have a much broader and more general strategy for protecting ourselves against those new viral pandemics.
Paul, what's your sense of this?
Well, as Tony is saying, in the larger sense and the longer term, we absolutely cannot just focus on coronaviruses for the reasons he's given, that many other existing viruses, Ebola, for example, remain very dangerous. And because, again, we can't predict in advance what kind of viruses are going to be problematic in the future. We've had several recent coronavirus events, as we've been saying, but it's instructive to recognize that prior to the emergence of SARS, coronaviruses were well known to infect humans, but they were only known to cause very mild cold-like symptoms. So no one was worried about coronaviruses. And one might have easily relegated them to something that didn't merit much study. The same is true for retroviruses before the emergence of HIV, for influenza before the 1918 pandemic. So we're continually surprised to seeing what we had thought were potentially innocuous virus groups turn out to be the source of major disruptions and disease and death. So the viral threats to humanity are in fact extremely diverse. And our response in building knowledge and antiviral tools also needs to be broad-based and correspondingly powerful.
So that's an important point, because currently our antiviral drugs, the few that we have, and certainly our vaccines, each one of them is very specific for a given virus. And yet we can't predict if there's going to be another virus that emerges and which virus might cause the next pandemic. And so if we're always having to react, is there a better way we can be ready to protect society in the future?
So that's a lot of what we'd like to talk about today are how our two groups are thinking about that, taking that broader approach. So the current approach, as we've seen, requires tailoring a solution for each new virus. So by way of analogy, it's as if this new virus is a locked door, and we have a new lock that emerges, and scientists are now scrambling to find the perfect special key that fits that brand new lock that we haven't seen before. And that key needs to be tailored to that one single virus. We can't go back and use an anti-influenza treatment necessarily, or Tamiflu, against this new coronavirus. So there's a real scramble and race to figure out what the solution will be to unlock this door. So if we step back, what virologists like Paul and I would like to think about for future viruses is how can we come up with a different model for opening that locked door, because we don't know what the lock will look like in advance. So again, to complete the analogy, we're looking for something more powerful in general, like a crowbar that can burst through the door, where we don't have to know what the details of that lock look like. We can use some alternative way to have a general effect, a general treatment against the next virus.
So yeah, a related perspective on this same goal is that for many common bacteria, we already have antibiotics that work against many pathogenic bacteria because they target conserved processes that are common to all of those bacteria. And unfortunately, we don't have such broad spectrum antivirals generally now. And to achieve a similar broad spectrum level of virus control, Tony's crowbar analogy, we're trying to build a much fuller understanding of how viruses work and identifying features and processes that are conserved across large groups of viruses. So by targeting these broadly shared features, we can develop broad-spectrum antivirals. The crowbar of Tony's phrasing, that inhibit not just one, but a whole group of viruses. Developing a collection of tools like that, a toolbox of such broad-spectrum agents, would give us a much better chance of dealing with new emerging viruses. There'd be a good chance that one or more of these existing broad-spectrum tools would work against a new virus, either individually or perhaps even more powerfully in combination with multiple such drugs.
So, Paul, that makes a great deal of sense, and I think everyone would like to see that. And yet, we haven't heard a lot about this kind of research taking place in a number of research centers around the world. And I'm curious, what are we doing here at Morgridge, and you in the Rowe Center for Research in Virology at the Morgridge Institute, to help develop these more powerful tools? And how widespread are such efforts around the world?
So, as noted earlier, our group is defining the molecular mechanisms of key virus replication processes. And one aspect of this work, among others, that has strong antiviral promise is identifying host cell factors on which viruses depend. So viruses have extremely small genomes, complements of genes, and for almost all aspects of their infection, they depend heavily on usurping, exploiting cellular functions. And in particular, we're interested in identifying host cell factors that many viruses require, and then spectrum antivirals. So using genetics and biochemistry, we and others have shown indeed that, as I say, that virus replication depends on numerous host genes. For example, for one advanced model system that we're studying, we've systematically tested the entire cellular genome, and we've already identified over 150 in many different functional pathways that are required for many different steps of viral replication. Again, some of these host functions and pathways appear to be required by many different viruses. So identifying and modulating those widely used factors and pathways can be a basis for broad-spectrum antivirals that would deny the replication ability of many different pathogens.
And that's really been a central focus of some of my research as well, is because we know that the virus depends so much on human cells and human proteins to do its work, how can we use computational methods to try to identify which of those human proteins, the host factors that Paul mentioned, are most promising to go after next as targets for these broad spectrum antivirals. And so just to go into a little bit more detail about that, the SARS-CoV-2 virus has 29 proteins, and proteins are the machines that the virus and human cells are using to carry out almost all functions. So there are proteins that are important for making our cells grow and divide. There are proteins that are used to help the virus replicate and make copies of itself. And through different types of experiments, scientists, virologists are able to map out which viral proteins can directly interface with or combine with or work directly with specific human proteins in the human cells that are being infected by the virus. So by tracking these protein-protein relationships, we can see that this particular viral protein has a direct effect upon some human protein. And we can also study which human proteins are being activated upon viral infection. And it ends up combining to form maps or what we call networks of different protein relationships. My group has put a lot of attention to thinking about how to use these networks, where we can see that the viral protein communicates with the human protein, which reacts with different human proteins, build up these relationships, much like the relationships that we have. If we think about tracking who we have our Zoom calls with every day, or who we communicate with directly or on the phone, that sort of social network is analogous to what we have with viral networks and viral human protein-protein networks. So then the computational challenges come in that we want to think about how to use that network information to prioritize which host factors, which human proteins have the most appeal as drug targets.
So I should jump in and just say that these network approaches that Tony's describing here have been very significant and important in our ongoing studies. One of our joint projects having to do with HIV, for example, had implicated a large number of host genes in certain aspects of virus transmission. But in most cases, it was not obvious what each of these host genes was doing for the virus. So this network analysis that Tony was able to provide by placing these genes in their larger context of protein-protein interactions and functional pathways, the direct and indirect connections of many of these genes to viral processes became clear, and we could see how they were contributing. The larger point that Tony began to allude to here is that genes and other biological components act in very complex systems, in networks. And you can't understand the function of these processes and their outcomes by studying just one gene at a time, any more than you could understand a novel by studying one word at a time. So these computational approaches now are very important in letting us envision and navigate the operation of these complex biological systems, and to an important extent, computation and is become a very important new kind of microscope for biology and biomedicine to understand the processes that we're studying.
So, if I understand this correctly, this is an important approach to developing broad antiviral drugs, because by targeting the human protein's interactions with viruses, you have the opportunity to interrupt those human protein viral interactions in ways that are less likely to interfere with the human protein's own normal function. And so it would be more selective for the viral infection rather than the normal human biology. And that's intriguing. Are there other viral features that you can aim at in developing new treatments?
Yes, I lost you there for a moment,
Oh,
Brad, but
okay.
I think I got the gist of your comments. I'm just going to step back and say that you alluded to an important issue here, which is, can we afford to target host proteins in an attempt to curtail the virus? And I just want to say that we have a number of proofs of concept that in the vast majority of cases we can do that in ways that are acceptable for keeping cells and tissues and organisms in good shape while we deny needed capabilities to the viruses. And then I believe you were also asking about are there other directions that complement this?
Correct. Yep.
In pursuing these goals of broader kinds of treatments. So yes, we have several such. One example would be that one of the most important processes in virus infection is making new copies of the viral genetic information, the genome, which is really the genetic payload of the infectious virus. That's what the virus is trying to deliver to the newly infected cell. So this process is so important that, to take the coronavirus as an example, nearly 75% of all of the genes in the coronavirus are devoted to this job of making new genome copies. So the virus clearly considers this to be a central or the central job of infection. Unfortunately, it's been poorly understood across not only for coronaviruses, but for all the other RNA viruses. So using revolutionary new imaging approaches, cryo-electron microscopy approaches that have been developed in recent years, we've been able to produce images at molecular to atomic scale and combine these with other approaches to show that RNA viruses, like coronaviruses, replicate their genetic information in exquisite nanoscale molecular machines or factories that are made up of viral proteins and host membranes. And by seeing how these factories are assembled and function, we're learning new fundamental principles that appear to be conserved across many different viruses. And so these are also now becoming targets for new, potentially powerful broad-spectrum antiviral strategies.
So let me just interject, if I might, with a question that has come up that I think fits with the topic of viral genome replication. And the question is, SARS-CoV-2 seems to be mutating more slowly than we might expect from an RNA virus. To what do you ascribe that slow mutation rate? Do you have any insights?
Sure. So I'll take a couple of, do this in a couple of steps. So one of the interesting features of coronaviruses is that they have the largest known genomes for any RNA virus. This is still very small compared to cells, but their genome size is, for example, 10 times larger than most, I'll say 3 to 5 times larger than most RNA viruses. It's in the range of 30 plus kilobases, 30,000 steps on the DNA double helix ladder. And because of that, it's been known for some time that they do a better job of protecting against errors being introduced during their replication. So they have a lower mutation rate than other RNA viruses like influenza or poliovirus or other viruses of similar viruses of that kind. And in fact, we know of special additional functions that they encode that provide some proofreading activity to reduce that mutational error rate. Having said that, it's worth noting that their error rate is still enormous compared to cellular gene replication. It's orders of magnitude higher than cellular replication. So even the coronaviruses with a low mutation rate for a virus still have capabilities to respond and change to changes in their environment, be that a new host, an antibody that a host has generated against them, or new antiviral drugs that we come up with. And we'll have to be watching that as we go along.
Very important. Thank you. Well, coming back to our overarching topic of being ready for new emerging viruses and how we can plan new technologies to develop new treatments. Tony, let me get back to that question about how is your lab focusing on developing new treatments?
So one of the things that my lab is most excited about right now is how we can use machine learning methods to be smarter about the process itself of developing new drugs or new treatments. So many, but not all of the drugs or treatments that we have today are small molecules or chemicals. And the challenge in developing new treatments is that there are simply so many different chemicals that could possibly be effective against a particular disease, or in this case, the particular virus that's threatening us. So there's this enormous search. We're envious of the people who have a needle in the haystack problem because we're out on a beach looking for one special grain of sand. So the question is, how can we really tailor that process of drug discovery, finding the right perfect chemicals for the new virus using the new computational technologies we have? So by analogy, maybe because I've been missing some of my favorite in-person dining experiences, I think about this as how I would explore the Madison dining scene. We're broadcasting from Madison here, and an uninformed default approach might be to download the menu from all the restaurants in Madison and order the first entree on every menu, let them come through delivery and start to see which of those I like until I finally find my favorite meal in my city. You can see that this is a very inefficient search process, because if I've ordered my 10th cheeseburger and I'm vegetarian, I'm probably wasting a lot of time and money figuring out that I really am just not a fan of cheeseburgers. So in that restaurant analogy, we can easily see there's different attributes of the food or the entrees we're ordering. Some are spicy, they have different ingredients, come from different regions of the world, and we can use those properties to learn rules in our minds very easily about what sort of things we like, and we could use that to guide the process and say, well, I really like Thai food, I should start ordering all the menu items from a Thai restaurant. I should really specialize for this one purpose. That's essentially what we're trying to do with drug discovery process, except the properties of these small molecules or chemicals that are candidate drugs are simply much more complicated, and there are so many candidate chemicals that we can't do this in our head. So the machine learning tools are trying to look at those same properties and say, well, what is it about these chemicals that suggests that they're going to be possibly effective against maybe this particular host factor, if we're thinking about targeting the human proteins with a broad spectrum antiviral, or in the shorter term, make them effective chemicals against some particular SARS-CoV-2 protein if we want to try to come up with short-term antivirals in that direction.
So I think that those approaches that both of you have talked about, excuse me, certainly make sense in a long-term approach to virology. But I think a lot of people in our society are saying, well, that's all very well and good, but look what's happening now. And people may say, why are you continuing to focus on a long-term approach? And well, let me ask you to think about how the COVID-19 pandemic demonstrates the need for such a long-term approach.
Well, it shows that both the human and the economic costs of viruses absolutely demand dedicated, expanded efforts to understand and control these enormous threats. So, as everyone knows, in just a few months, COVID-19 has already claimed in the US 100,000 lives, more than the Vietnam and Korean wars combined. It's cost us trillions of dollars, and it threatens every aspect of our economy, from small businesses to major industries like airlines. So, underscored by HIV, by Ebola, by many other recent virus outbreaks in the last decades, this is a pivotal wake-up call for public health. As we've said, we both need to deal with COVID-19 immediately, and we need to recognize the obvious lessons that viruses represent one of the greatest threats to public health, and also one of the greatest threats to global economic and security interests. So just as we as a country have responded to other potentially devastating challenges, and we're struggling now to collectively grow in response to crucial social issues, in this case also, we need to step up with a vigorous and dedicated long-term plan, fully commensurate with the scale of the issue. So if we don't want to be living under the continuing threat of another COVID-19 or the next HIV or the next even faster spreading Ebola virus strain that might be more deadly, we need a broad-based space race type of approach against viruses. To do otherwise would be irresponsible. And the cost will be small compared to the trillions of dollars that we've already lost to COVID-19. Now, fortunately, recent advances in virology and in associated fields provide very exciting foundations, as we've been saying, for powerful next steps to understand and control viruses. So we can be very confident that investments in these critical areas will pay off well for society.
Yeah, and certainly both Paul and I are maintaining a big picture long-term approach while simultaneously asking ourselves, what can we do with the tools and the skill sets and expertise that our labs have? So both of us have taken this opportunity to do whatever we can to have short-term effects and use our existing skill sets toward this particular pandemic to try to shorten the duration any way we can. But it really is not lost on us that we need to be prepared for the next pandemics and need to be thinking about these alternative approaches so that we're not always caught in this scramble, and that if another COVID-19 like pandemic were to strike, we're not going to be forced into physical distancing or social distancing, as it may be commonly termed, for quite so long. We're not going to need to have the same sort of societal disruptions. We're not going to be able to extinguish viruses worldwide. That's certainly not the goal, but we're going to be ready so that the consequences aren't so dire if these long-term research plans succeed.
Great. Thank you very much. So we got a couple of questions in that I think are germane to some of the things we have been talking about. And so one of them has been, do you believe that the same principles that would enable discovery of a broad spectrum antiviral drug would also be viable for creating a broad spectrum vaccine? So whichever one of you feels that you'd like to address that.
So I'll jump in little. So there are efforts underway to expand the efficacy and range of effective vaccines with influenza, where currently the influenza vaccine has to be reformulated on a yearly basis because of rapid changes in circulating influenza strains. So what's being done there is similar to the concepts that we've been outlining in terms of looking deeply at influenza and identifying features that are more conserved, that don't change with the rapidity that some of the protective surface antigen features are shifting on, and targeting those. In terms of even broader efficacy of vaccines, there's a lot of interest, including on our part, in looking at how we can enable different levels of the immune system, including responses that can have very broad effects. There's a strong and parallel that we hope to grow in future with cancer research, where immunotherapies in cancer now are a very important area of advancement. And what is being learned there can be likely advantaged to generate further renaissance in antiviral immunotherapies, just as advances, for example, in HIV therapy, the advent of cocktails of combinatorial therapies against HIV has been very instructive for generating strategies to better treat certain cancers. There's a lot of crosstalk between and a lot of fundamental similarities and crosstalk in terms of virology and cancer research, and we hope to continue to exploit that.
And Paul's answered that quite well, but I think it bears repeating that one of the reasons that targeting human proteins and host factors is so appealing in the search for broad-spectrum antivirals is that the virus has no control over the human genome directly, at least upon infection. So when we think about viruses mutating and viral evolution, making vaccines less efficient, that isn't the same concern we would have if there were therapeutics targeting human proteins that we might hope would have longer term, more permanent effects or be more durable over the long term.
So that brings up another question, and again, it refers back to the immune response. Excuse me. And we often hear the term herd immunity, and a question has been submitted about is there any validity to the assertion that herd immunity can be effective? Or are we going to have to depend upon a vaccine and the development of each individual's immune recognition of the virus? And of course, the two things are connected. And if I might ask one of you to comment on that.
So I think it's important we draw a distinction between virologists and epidemiologists. So I would say that epidemiologists and other types of scientists have studied this question of herd immunity very carefully over the decades, and then thought about how those core concepts of herd immunity apply in this particular pandemic. And if you ask 10 scientists the same question, you'll get 10 slightly different answers. But there's definitely a consensus opinion that's formed in that if you model what racing toward herd immunity would look like, it would be simply catastrophic, that's not a viable approach. So when we were talking about flattening the curve earlier on, it's related in that it's not simply the number of individuals that get affected that's going to answer this question of when do we have herd immunity and what's the trade-off between herd immunity versus individuals' immune systems versus vaccinated individuals, but it's how many individuals are infected at a single point of time, and how are hospitals and healthcare systems overwhelmed if we do have rapid spread of infection through our communities. So the consensus has been that we really do need to do what we have in our power as a society to avoid spreading this and give scientists and pharmaceutical companies time to develop and test these vaccines so that we can distribute vaccines once they've been shown to be effective widely and attack it in that manner. Paul, perhaps you have a follow-up.
I think that's well put, Tony. Obviously, we're balancing different needs that society has. Hopefully, as we're going forward, we are learning more about what are the most important transmission risks and how to better protect against them. And as has been widely noted, we're ramping up our ability to do testing and that kind of information, knowing what the level of infection actually is in a community, where there might be hotspots. All this kind of knowledge can help us to better control the infection by practical means while we're working on vaccines as the benign way to achieve broad population immunity, as opposed to the more devastating path of natural infection.
And I might add that there is an overlap between herd immunity and vaccine-induced immunity. In that, so for members of the audience that may not be fully acquainted with what herd immunity is, it's when there are so many people in the population that are already immune that the virus doesn't get passed on. And I think that that's important. By way of illustration, if we have a population where there's very little immunity, and you have 10 people in a room, one of whom has the virus, the chances are that all 10 of the people will eventually catch it if you spend enough time in the room with the infected person. If 9 people are already immune and one person has the virus has nowhere to go, because every time it tries to jump to another person, that person's immune system will prevent the virus from establishing an infection. The result is, in the first setting, you now have 10 people that are infected. Everybody goes out and infects other people. In the second example, the virus just doesn't spread. And that's one of the reasons that herd immunity is so important. And it actually doesn't matter from a virus transmission perspective whether that immunity is established by vaccine or whether it's established by true infection. The problem with true infection is, as Tony pointed out, a lot of people will die along the way. In order for enough people in the society to manifest immunity, a lot of people will end up being terribly harmed by it. So the vaccine is a more effective way to get enough people in society immunized so that the spread of the virus is stopped. I think that's a very important thing. We've been talking about the importance then of understanding fundamental mechanisms to block a future outbreak. Could I ask each of you for one example from your own work where your work has benefited from a previous fundamental discovery that has allowed your current work to move forward? As an example of why ongoing fundamental research is so important.
Sure.
So I'll lead if Paul needs a moment to think of his favorite response. He's had a longer career, so he has to think through many more transcendent discoveries. So interesting, a lot of the computational methods that my group is now working on, specifically to look at broad spectrum antivirals, were really inspired by some work with boring old single-celled yeast. So we have a collaborator who happens to be at the University of Wisconsin-Madison, who I never met, but was doing really fundamental work, understanding how these single-celled yeast respond to different types of environmental stresses. So they did these interesting studies where you grow the yeast for a while, and then you make it much colder, or you make it much hotter, or you dump some salt in their environments, or you deprive them of the nutrients they need to grow. And this just generated fantastic data where you could track over time how different elements of the yeast cells were turned on or turned off in responses to all these different stresses. And that really inspired me during my graduate training and led to some of the computational methods that we developed, first to better understand those yeast responses and now to understand human disease.
Great, thank you.
Yeah, I can echo that with comments that there's a tremendous number of examples where our work is dependent on technologies that, of course, were developed elsewhere in painstaking steps by many groups. I've already referred to the cryo-EM imaging, which is so advancing many studies in biomedicine right now, including work that we're doing and others on campus. Other examples exist in major genetic technologies that are now allowing us to test genes in systematic ways and to engineer genomes in powerful ways, both to ask experimental questions and also to help to generate antiviral states. Yeah, it really, in a scientific sense, takes a village of contributions to make all of these things happen. And it's one of the great privileges in science to be a beneficiary of so many diverse contributions worldwide.
So as we get close to the end of our session, I do want to highlight an important question. It's something that we often do when we explain a biologic process, is that we say that, for instance, the virus is acting with intent. We talk about something, making it seem like the virus is planning and it's doing something because it wants to do something. Well, when in fact, the things that happen are random events, and that the result of those random events either turn out to make the virus more hardy, and therefore that event persists, or that characteristic persists, or it makes the virus less hardy, and the viruses with the less hardy characteristics don't persist. Can you comment on that? And we all do this because we want to get the idea across about change and the sequelae of that. So, Paul?
Yes, so we probably are guilty of anthropomorphizing these systems in some respects. You were just referring to, in this situation, Brad, and we referred earlier, mutational randomization as an extremely important aspect of viruses. And what you say is very true, that it's simultaneously clear that a high mutational rate is advantageous for the virus in providing the ability to adapt to many situations, but simultaneously it carries a burden, severe burdens of inducing lethal mutations in many of the progeny viruses. I referred before to the enormous mutational rates involved. Most viruses that replicate through RNA intermediates, RNA viruses and retroviruses, the mutation rates turned up so high that they're just balancing on the edge of collapsing into genetic catastrophe. And so the burden of deleterious mutations are indeed very great. And it does look to us like many of their genomes are non-functional as a result. At the same time, we now have mutants of these viruses that have greater fidelity in their replication. They make fewer mutations, and they are, even in short-term infections, less fit, particularly at the whole organism level. So it appears that these mutational capabilities are important even for steps as simple as moving from one tissue to the next, that the virus will adapt in moving from one kind of cell in a mouse or in a human to another kind of cell. Quite intriguing how these systems have organized themselves to be functional in ways that we might not have thought to do, and to expand their general capability and their access to different sequence spaces.
And of course, they attain those characteristics by chance. They didn't plan it out.
Yes, we could open the door here on a complex set of genetic discussions, but this may not be the time for that.
Tony, any comments on that before we close up?
I think Paul's covered everything that I might say on that topic.
All right. Well, we're reaching the end of our time. I want to thank everybody for attending. A couple of points that I want to make. I hope it's evident why it's so important to do basic fundamental research and really understand the underpinnings of how things work. Because once you figure out how something works and you've got the information right, you can be much more targeted about your approach to it. And unlike the viruses, we can actually be purposeful and work out things that work. to our advantage. The second thing is this virus is going to be with us for a while. And it is important that all of us continue to be attentive to it and that we continue to do our part in society to help us get past it. And that, I would say, will take many forms. So one, you should continue being curious and you should continue learning things. Number two, if you have an opportunity to participate in a study that is investigating preventative therapy or a treatment, I would urge you to think seriously about participating. Number three, if you find the work that any institution that's doing is important and interesting, you should think about learning more about it, supporting them in ways that will help them. And then also, please realize that most of the funding for ongoing research in this and all sorts of other important areas comes from the federal government. And it's important that your elected representatives hear from you and that they know that ongoing research is important to you and it's something that you feel should be supported. So there are lots of ways that everyone in society can have a role in not only getting the better of the current virus, but preventing and appropriately fighting whatever comes out in the future. So I want to thank everybody for attending. And I hope you found this as interesting as I have. I'll tell you that we're planning on having another webinar in July, so please be alert for news about that. I'd also like to tell you that you'll have the opportunity to sign up for our electronic newsletter, and we'll make available to you a link to a video recording of this. So if there's any of the content that you want to go back and revisit, we hope you will do so. So thank you all very much for attending. We hope you learned a lot, and we hope we'll see you again on another one of our presentations. And Paul and Tony, thank you very much. And I want to thank Gabriella for all of her hard work behind the scenes. And most of you didn't get to meet her, but we owe her a debt of gratitude for making this happen. Gabriella, thank you.